I see bottom in sight. Yeah, looks like a silty sand on rock. We're going to settle in, get closer, let the ROV settle down, and our pilots to get comfortable. And then we will start zooming and get a good idea of what lives here. I think there's some gorgonians I'm seeing, some long stick corals or whip corals, I think. A couple of fan gorgonians. It's pretty rugose, much more rugose than I was expecting. That's a good sign for biota. I think there might be some white sponges here at 3 o'clock. Maybe, see the tube sponges here?
Yes.
That's pretty cool.
Little euplectellids.
Mm-hmm. Maybe a bamboo coral at 9.
Wow.
That's neat.
It's looking good.
So there should be a slight slope here in the 50 degree-ish, maybe more. It looks much steeper than that when we're staring directly at it, but the overall slope should be pretty close to about 50 degrees right in this local area, but once we start moving, it'll get steeper.
Mm-hmm.
Yeah, it looks like that silt's probably coming down from the upper platform, from the plateau above, right, where it just kind of...
From the plateau and then also from the water column.
Mm-hmm.
So we're on a little promontory. 3,647 meters.
Yep.
I'll note as we're getting settled here, you know, this is pretty deep, as you said, more than 3,600 meters, and there's a definite depth stratification to the coral species, and likely sponges and other deep sea communities. And so the things that we see down here tend to be different species from what we see in our shallower dives. And they're, for the bamboo corals, I only know of 2, maybe 3 species that are known from this depth. So I do think it'll be interesting as we're looking at them to see that they are different from what we've been seeing on the previous dives.
Thanks, Scott.
Great.
Does anyone know what time we took our 500-meter sample of water?
I do have that information. It was 9:16 EST or 13:16.
13, there you go.
I should probably revise my last statement to say that there have only been a few species described from this depth. It doesn't mean that there are others that we don't know about yet.
Sure.
And specifically bamboo corals.
Roland, my C-tube records in GMT, which is why, since you asked. So if anyone listening, C-tube is the data management system we use to log critters and habitats. Yeah, annotation data.
And one of the nice things about this tool is that it can be used to annotate live, but then also after the fact, it incorporates a recording of the video from this expedition, and people can continue to annotate it as we learn more, or as different specialists get to spend time with the video. Right,
so it's like a super collaborative, and it's data managed, so like a lot of marine biologists use worms or the World Register of Marine Species, so they can actually look up the species in the worms database directly. So it standardizes it so that all the naming conventions are right. And then when Worms updates its taxonomic information, which I'm pretty sure they delight in torturing me with, it automatically updates the data inside C-Tube. So it's actually it's a great tool to have, and I love it. It was invented by Mashkor. I don't know Mashkor's last name.
I'm not sure.
Yeah. So I think he'd use this for his PhD project, actually.
As an editor for the World Registry of Marine Species,
it's your fault.
I can confirm that we are not trying to torment you when we update the taxonomy as papers are published.
It kills me.
Yeah.
Here's some cool. Is it Euplectella? These white tubes. This one with branches over here at 9, but most of these are the single tubes.
Don't think Euplectella branches, so...
Definitely hexactinellids, at least.
Yeah, they really look like hexactinellids. They, not to get ahead of ourselves.
I know, but I gotta look at it.
No, I mean, they look like great things to have in the bucket.
I think down at the bottom at 6, there's a flytrap anemone.
It's
looks like some pink stuff, probably brittle stars.
Or is that a star? Dead center?
I'm not sure. I'm not sure if it's a star or brittle star. I'm going to go with Phylomechina dermata.
I'll trust you on this one. It's Malik Mashkor, by the way.
Thank you.
Or Mashkor Malik. Yeah, one of the other things we didn't mention about this annotation software, C-Tube, is that it also has all of the environmental data that's collected. So when you make a taxonomic annotation on the video, it's right there associated with oxygen content, salinity, temperature, etc. So that's really nice, as well as the timestamp. Ooh, is that an anemone?
Yeah, this guy right here. Yeah, I think that's a flytrap anemone. Yeah.
Hormathidae.
Yeah. Now you're going to get yelled at for that.
Oh.
Because there's like 15 different classes of flytrap anemones, and you can't tell them apart unless you pull them up. Because I always want to call them that too.
Oh, that's good.
But they yell at me. They get yelled at for that. So, give you a heads up on that.
I don't know if it's yelled at.
Corrected. I get corrected.
Yeah.
No one actually yells at me.
That's the nature of community science.
Now these look flat. They're not tubular at all. Some of these white ones.
Great.
These are really common, these white. They almost look like staghorn coral or something. They're definitely a sponge, but they're really flat and then forked. Lobate with lobate branching.
Yeah. I have no idea what they are other than glass sponges. They're very characteristic of this site.
Yeah, they're really common. They'd probably be worth collecting, yeah? You think?
Yeah, if there's an easy collect opportunity, I think it would be nice.
Yeah. Yeah, that's fine. If you can get a good shot of some of these flat, low bait, tall sponges, and even that white cream colored one too at like 6 o'clock ish. But go for that white one in front of me. Just snap zoom, right there. Yeah, flat one, the flat lobe. We don't know what those are. Looks like you've got dead ones too. See these brown? They're browned out, the same shape.
Yeah, I think so. Good eyes. When they're dead, then the sediment accumulates on them. This is a good choice.
Oh, neat. Some kind of crab or something on there? I don't know what that is.
Wow.
That's crazy.
I do not know what arthropod that is.
No, either do I.
That's an isopod.
Isopod.
Is that an isopod?
Okay.
Wow.
He's very funky.
He's really cool looking.
Very roachish.
So he's squished rather than squashed. Isopod.
There's some in either inside or on the back side of this. I can't tell if this is a tube or a flat, like a flat blade.
Mm-hmm.
I think it's a flat blade that's kind of scooped out on the other side.
Mm-hmm.
And I wish I could remember the name of the group of these sponges, but can't seem to remember many names these days.
I can confirm that
Ooh.
that was interesting behavior.
He's very weird.
It is
Here's
really
some
neat.
cool gray one over here at 4.
Yeah. Yeah.
Oh, no, don't apologize. Don't apologize. You do whatever you need to do.
I can confirm that there are no glass sponges of this form in the Smithsonian, which
Oh,
has pretty much the best glass sponge collection in the world.
Yeah, we're going to, if we can get someplace safe, we'll grab one of those. This one's really cool at like, see, 8, 7, 8 o'clock. It's got lots of lobes.
I wonder how common that isopod is going to be.
It looks like bamboo corals here at 9. Possibly a single-stalked one at 6. It's very rocky and robust. There's lots of rugosity. Not a lot of fish. So in the mesophotic area, we have lots of rugosity like this. We usually swarmed with fish. So we're just way too deep for that. There's a cool fan sponge, dead center. Yeah, so we're crabbing to the right because of the currents. So the ROV pilots are trying to settle it. But that Cirrus might just take us that way.
Right.
Here's a cool fan, like stalked fan or cup at 7. And now we're looking out over the side view into the void. Awesome. So this, whatever this thin fan hexactinellid sponge is the most common species down here.
Gotcha. Yes, definitely.
It's everywhere.
It's more anemone there.
Another, yep.
Something.
Yeah. There's 9 o'clock. It looks like an ophiuroid. There's more in the front. There's one at 6. There's one at 3.
Straight over the edge of the rock.
We also have this same white ball sponge with the large oscule that we saw a few minutes ago at 4. That's probably hexactinellid.
I'm thinking,
Think
yeah,
so?
a large sort of barrel-shaped.
And we've got, the ophiuroids. So the hot pink 5-armed brittle star. There we go, moving in a little bit. So here's that white sponge we were looking at.
Oh, very nice. Almost certainly that's definitely a glass sponge.
Yeah. And then we have some more hexactinella-like sponges in front of us, maybe possibly dead ones.
Yeah, or little dead stalks with other smaller sponges growing off the side of them.
There's also a really cool cup sponge right off screen. Right here. There you go. See the?
Oh, yes.
See that? Like it looks like a.
I've
like a bowl.
seen that before. It looks like a sponge that Henry Weisswig described from the West Coast.
Oh, there's another white or cream-colored ball. It might be related to this one over here. It's like 4. It's a lot like more robust, right, than those white fan low bait ones. I've got
more
some
dense, right?
pink coral at 6 o'clock, a gorgonian of some type, maybe a black coral. Look at these slabs of rock. They're crazy. So like 2 or 3 meters, maybe even 4 or 5 meters wide. Those slabs. And they're rounded, look like they've eroded on the edges. Here's another one of those bowl cups on a stalk. We're going to do our on-bottom water sample.
So we're coming up on our second water sample collection.
There might be a Chrysler type gorgonian here at 9 o'clock. Looks like a fan, or a feather almost. When we get closer to it, it'll be more bushy. But I believe that might be a Chrysler.
Dark in color.
There's another thing I don't recognize here at 6. All right, that was water sample 2. Awesome.
Fantastic.
It seems like that Niskin bottle 2 is a little fussy. Doesn't always fire when it's supposed to, so. If you feel comfortable collecting one of these white low bait fans, that would make me super happy. Yeah, there's actually one right at noon that's quite large. I don't know. You judge. You make the right, you pick one, but. That'll work too?
Yep.
Yeah, go for it.
All right.
Yeah. This looks like a dead Rosilidae, right? With that rounded top.
I would
Here
not
at
be
3
surprised. Rosalidae.
Yeah, and if you have to bail on the sample, just no big deal. Okay. Mm-hmm. Okay.
So this sponge that we're collecting, it's would be novel for the collections at the Smithsonian, which have a rich glass sponge collection, and then also they're very characteristic of this site. So
Yeah,
we
they're
have
super
multiple
common.
reasons to
Yeah.
to collect this.
So they're about 10 centimeters long. Oh, whatever you're comfortable with doing,
Yeah,
you
whatever.
can do. We're not going to fuss. Yeah.
That's all right.
So I would describe this as about a 10 centimeter flat lobate. Like cluster of lobes, really.
Yeah, it's like a branching fan.
Mm-hmm. And the best we can do is hexactinelle it, I believe. All right.
Really abundant.
Yes. I'd say most common species I can see.
Nolan Barrett in the chat room says that this looks like a Tretoplura.
Tretoplura.
Yeah. So.
Yeah, pilots, this should be very soft, very fine, easily broken.
Not a problem. Thanks very much for the attempt. I appreciate the difficult conditions you're working under. Mm-hmm. That's not a problem. I sense new opportunities on the horizon. It's so much of them. Yeah. It's really quite the landscape.
Yeah, it's a lot less steep than I was expecting. We were aiming for a more flat area when we landed.
Mm-hmm. Wow, look at the size of that large one there. About 10 centimeters across based on those lasers.
Mm-hmm.
Maybe 30 centimeters high. What's another one of those brittle stars perched on top of the sponge?
Mm-hmm. They all seem to be growing roughly the same way. They kind of line up like fan facing, right? Probably the incoming upwell current off the shelf.
Yeah, facing the deep, right?
Mm-hmm. There's a really nice one at 3. I'm sorry, 9.
At 9, yeah, look at that. Looks like a big mitt.
Mm-hmm.
And this density of organisms down here at over 3,600 meters, below 2 miles from the surface, where there's not a lot of nutrients. It's really remarkable. We have Asako Matsumoto from the University of Chiba commenting in the chat room about how incredible this diversity is to see here.
I think there might be a Demo sponge here at 9, 10-ish. See that yellow ball? Or cream colored ball?
Sure do.
This is a different hexactinella too, right above it. It looks circular, tubular.
Yeah, maybe.
All right, so we're going to try and start collecting sample 3 again.
Yeah. Awesome. There goes the claw.
The claw.
I mean the arm.
Well, I mean, that's a claw.
It is a claw.
And it looks like there's a dead one next to it. It's the same shape, but it's a brown color. So it's probably been dead there for a bit. It does look like a mitten, though, doesn't it? Or like a scuba diving glove where their 3 fingers are together and your pinky's out.
That's right. Can you count with those 3-finger gloves?
Yeah, yeah, to 3. Yeah,
Ooh,
really
it's
fine.
really
It's crispy.
soft. Cracked. Wow.
It popped right off.
So delicate. Such delicate work. Port side, great. So again, these taxonomy, the glass sponges, is really heavily tied to the morphology of their spicules and their skeletons. And so unless you already know what a species is, it's difficult to identify from the pictures. Unless it's something that's already very well characterized, because the gross morphology of re-evolves in different groups quite a lot.
Yes. It's just going to back away and then store
Understood.
it.
Is that oh no, just saw something falling. It must have been one of the dislodged sponges. Yeah, and no worries.
All right.
Yep, it looks good.
Yeah, you can see the rippling water coming out of the bucket. Temperature gradient. This is so stressful.
Yeah, that's right. Port, that's going to be outside.
Got him in?
And
Yay.
success.
Thank
That's
you.
so great. Thank you. Great work, pilot.
Yeah, that was awesome. Thank you, guys.
So,
I'm going to try and start moving a little bit shallower.
yeah, not a bad idea if we can move up a little bit.
Hey, who chimed in?
Yep. So while we're sitting here, because everyone wants taxonomic notes, right? that glass sponge, which at least tentatively is ID'd as Trichoplura, that's in a family Uncinetaridae, and that whole family name is based on a spicule shape, So it's something that we couldn't possibly observe, through the video. It's something that can only be done back in the lab to know.
It's a very unique shaped sponge. It's like, lobate and taller than it is wide in most cases.
Mm-hmm.
Thin, and it was really crispy when we touched it. So it broke very easily when D2 reached out.
Snap those glass spicules.
Yeah, right. We'll give you a recap now since we're waiting for the ROV to feel comfortable. ROV pilot, this is science. Let's see. I don't know if he can hear me. He might have me turned off. Okay. Yeah, I was going to say, if we can start moving up the wall, that would be awesome. But at your discretion, of course. Yeah. Okay, awesome. So just to give you a recap, we're about 274 miles from, I think, Jacksonville, Florida, roughly, along the Blake Plateau. The Blake Plateau is about 127,700 square kilometers, and it ranges in depth from about 450 meters to 1,200 meters, the deepest part being the southeast corner of the Blake Plateau. The first survey of the Blake Plateau actually occurred in around 1881 by the U.S. Coast and Geodetic Survey steamer, the Blake, which is the ship. It made about 13 soundings and sampling lines across it, so it's really minimal data, but that's where the Blake Plateau was actually named after in 1881. For the most part, they really didn't know much about it until like the 1960s, when a few geologists, primarily George Stetson, who actually I think Stetson Bank is named after, and they actually mapped this deep water area. And Stetson Bank is off Georgia, and it is named after George Stetson. And I believe he's a Woods Hole scientist. So, yeah, we're way offshore.
Yeah, we're on the Blake Spur, so a little spur that sticks out off of this Blake Plateau.
Mm-hmm. And the spur is, this like protrusion, if you will, on the edge of the Blake Plateau, which drops out down into the Blake Basin, and we are currently 3,650 meters underwater, which is roughly 8.5 Empire State Buildings stacked end to end. Give you an idea how deep underwater we are. 2.5 miles, you said?
2.3 miles. Yeah.
And this ledge, from top to bottom, is taller than the Grand Canyon is deep.
Yes.
To give you an idea, if anyone's ever been to if you haven't been to the Grand Canyon, I highly recommend. It's beautiful. But that's really remarkable that this features bigger than that.
Yeah, this wall is, I think it's 1.3 miles tall. This wall.
Just the wall from top to bottom, from the plateau to the base.
Yeah.
Crazy. So we're gonna see a tiny little matchstick size of this humongous wall. That's how science is, right? You just sample small sections and kind of try to expand your knowledge of the biota in the area and collect some data, and then use that to kind of make, hypotheses about what lives here. And if likely this biota is here, it's also laterally in the same depth across most of this feature.
Yes.
Most times.
Yeah.
More data points are better, but,
Of course.
There was only one dive in the area that went all the way down to the base of this, and that was by the Alvin. And I do not know what their data showed, so we are flying blind as far as I'm concerned. This is all exploration.
That's okay.
Yeah, that's what we're here for.
Exactly.
It looks like parts of this wall are almost 90 degrees. They're just straight up and down, according to our multi-beam. And it looks like the main species here is this white fan that we just collected. It looks like a little mitten, a little flat mitten. Awesome. Thanks.
So we're going to have a pilot change here, and then we're going to start proceeding up the cliff wall.
And that last sample went into the port outer, correct?
Yes, port outer. At 15:52.
That's the lunch bell.
Is
someone else monitoring this, or are they going to start?
Yeah.
And I can always go back shortly.
It's interesting. There are a lot of comments in the chat about the currents that must be experienced here. We know there are strong bottom currents that influence this region, but the delicate nature of these sponges would seem to suggest that the current doesn't get too strong in this area, or it would knock many of them down. Taking a peek up the slope here. So we're going to start to make our way up. We're at 3,646 meters presently on a wall of the Blake Spur. This wall seems to be continuing to be dominated by this one glass sponge that we've made a collection of. I see a couple whips there. so. Excellent. Nice close-up view of this. For me, that's an octocoral, a bamboo coral. You can see the nodes. You can see the nodes I'm referring to are the little dark portions along this stem. It'd be great if we could get a zoom in on maybe one of these smaller white sponges. There's one straight in the middle there now. Yeah, that's the one that we've collected. Maybe just above that on the rock. Yes, thanks. Get some great footage. I really don't know what it is, but maybe someone will. Thank you. I think we can come back out and look for something new. Ooh, there was another glass sponge. It was just to the left and behind that rock on the bottom. Had a completely different morphology. It's pretty dead center. And then now just a little bit up from there. It's, yeah, a little fluted guy. Maybe Pharea, I think. Great. Yeah, I think that's likely Pharea or maybe a Euretid, but thanks very much. So right now we're just trying to get nice zooms on some of the diversity of the larger organisms here. You can see from this view how densely how abundant first this fan sponge, which seems to dominate this area, but then in and amongst them is a really high diversity of other sponges, sort of a spongepalooza thing going on here. Yeah, that would be great. Let's take a look. I think that might be a brittle star. Ah, I see what you're coming in on. Yeah, that looks like rust. So there was a little crab to the left if you would like to check that out. Just in that nook. Yep, you're pointing at it right now. Yeah. It's covered with armor, sponge armor. I think that's a hermit crab. Does it have a small shell on the back?
Looks like he's wearing an anemone.
Yeah, he's maybe has a pom-pom anemone. So he's a whole ecosystem in and of himself, you know, carrying sponges and anemones. And I would imagine if it does have a shell back there, that that has even more organisms living on it. So crabs that associate with anemones like this, it's been shown pretty clearly that the ones that have been studied closely, that both organisms benefit, that the anemones get food waste that that the crab, when the crab's eating, and then the anemones also serve as protection from predators. Thanks for that zoom. So we could come in on those stars on that rock that are 10 o'clock. I don't think we haven't had a close-up of them yet. These are brittle stars, ophiuroids, climbing up on dead sponge and on the rocks there. It is a sponge paradise for sure. So it's been dominated by this one blade-like glass sponge, which we see there left and right. But then there are tens of other species in and amongst them. Most of them glass sponges, but also we've seen quite a few sponges that are from the demo sponge group. It seems to be, is that a white crab there? All right, I guess that's I think so, a Galatheid. Looks a little, definitely not like the ones we are seeing up on top of the plateau.
How'd I get on mute?
I think that's okay. That's not a collection possibility, is it?
Oof.
Yeah. If it could be sucked up, I think it would be a nice collection, but it's definitely seems like something new.
It's on the move.
It is on the move. Thank you, pilots, for considering that request. Yep. We'll see. He's pretty mobile.
think it might be wise to our game here.
Yeah. Is understood. Understood. Thank you for considering it. Yeah. I think, yeah, we can go somewhere else. This is... Oh, what is that?
Is that wood?
It looks like wood.
Huh.
Decaying wood. Probably full of interesting worms digging it out. Oh. So we're slowly working our way up this wall. And we'll get in position for some additional zooms to get a close-up look of what's growing in here among this fantastic spongescape.
Oh, the puns.
And we have our co-biology lead dropping back in.
So we made about 100 meters headway in the vertical direction.
And we were going to attempt a collection on a squat lobster, a white one, very interesting one, but he was wise to us and
took off.
Ran into the cracks and said, "No, thank you."
Yeah. So these white mitten sponges are really the most common
Yes.
thing still, yeah.
Yeah. You missed, This is a fantastic spongescape, don't you think?
Oh, nerd jokes.
Yes.
Looks like a piece of dead bamboo coral.
Oh, yeah.
9.
Yeah, I'm looking forward to the next opportunity to zoom in.
So these rocks are like jumbled, right? Like a jumbled pile of eroded, right?
Yeah,
Maybe
looks like-
Yeah.
10 to 30 centimeters, and then some up to like 2 or 3 meters wide.
Yeah, looks like some fractured cliff faces
Yeah.
coming down.
Ken Sulak in the chat is commenting how these rocks would likely create lots of eddies from the current and little localized that would sort of hold in the particulate matter.
It's fairly rugose. Lots of hidey holes, places for
Like the one that
animals.
squat lobster ran into.
There might be another squat lobster on that bottom right rock right there. See maybe some legs sticking out.
Oh yeah, look at that.
Good one.
It's a little guy.
Yeah.
And a mirror?
Yeah, an animurine of some sort.
Flattened out a little bit here. It's a little less of a slope.
Right.
Still into the abyss on the side, so just locally. Looks like a huge rock in front of us, maybe 2 or 3 meters wide.
Let's go look. The ophiuroids, Chris Ma identified them as that we've been seeing climbed up on top of these sponges, Ophioplinthaca. Oh my goodness, good name. Ophioplinthaca.
Those are the pink ones we've been saying are
Yeah.
common? Okay. I'm going to be copying that out of the chat room because there's no way I'm going to spell that right.
I got it down for you.
Mm-hmm.
Do you want?
Looks like another solitary stalked Gorgonian center screen. I don't know if we've gotten a good zoom on that one. Maybe a bamboo coral? No, I don't think it's a bamboo coral. I'm not sure what this is. Oh, yes it is.
Yep, there's a black ring.
Yep. Awesome, thank you. Yeah, it looks like a jumbled pile of rocks. That's what this wall's mostly made up of, at least in this area.
Yeah, slope failures coming down. Yeah. Some sediment mixed in there, which is to be expected.
Yeah.
Especially this deep.
And right, we were on top of this plateau a little further north earlier, yesterday and the day before, and it was mostly sediment. So I wouldn't be surprised if this is just coming down, being blown off the top of this plateau.
Yeah.
And along the wall. That looks like a Chrysler, maybe?
Or Metallogorgia, maybe?
I don't know.
Only if Scott was here.
I know.
Always one of my favorite corals. Just so stunning and beautiful.
Yeah, they're really
So
nice.
delicate.
Really fine branches, really bushy. Some nice pink polyps. Awesome. Maybe an amphipod there?
On the left side?
Yeah, at 9 o'clock.
Yeah.
He's got his hot red color in his face. Or on
Looks like
his
there's a
forward
few of
part.
them.
One down near 6 o'clock. Maybe one white one at the top.
Yeah, it looks like 1 or 2 at 12 o'clock. Yeah.
So maybe 2 species of amphipod.
Is he trapped or is he just sitting there?
I think they're just hanging out. It's their little underwater tree.
Oh, there's a few of them. I see more of those little white ones. And those bright red ones look like there's at least 3 of those. And blowing in the breeze.
There's a shrimp in the center there, isn't there?
A shrimp, you think? These flattened out amphipods, that's what they look like.
So Nolan Baird and Tina Molitsova are discussing this on the chat, and sounds like it may be a baby Metallogorgia.
Oh, nice.
Which is quite beautiful.
Yeah, and you can really see the direction of the current right there, from right to left, blowing those polyps.
Mm-hmm.
Now, is this something that's recognized? Is this?
Yeah, they were saying maybe Metallogorgia, but a juvenile.
Mm-hmm.
Right. Yesterday we had the ones that were on the long stem, and at the top was like a flower, where the polyps were growing. And Scott had mentioned that when they're young, they actually grow branches all the way up their main stalk, and then when they get to a certain age, they drop those side branches and then just have that flower head at the top.
Mm-hmm.
So the thought process on the chat was that it was a baby Metallogorgia. So
Interesting. Also, some chat comments about how clear that current appears because of the way it's moving the polyps around on the octocoral, but then these glass sponges are just standing up to it, not flickering around.
Right.
So they have quite a lot of, even though it snapped easy when we did it, they have a good strength against the current.
look at this
That'd
huge
be-
rock. My goodness.
Maybe rigidity, that would be.
Yeah, like the oak tree.
The good one will blow in the breeze or the current, right? So
Yeah.
it doesn't snap. They seem pretty strong.
Pelagic critter. 12 o'clock.
Maybe another dead bamboo coral at 9 that's sparsely branched. And then we have a few more of those single-stemmed ones. So we're coming up this humongous rock formation. It's very jagged.
Wow.
Yeah, I wonder if that fell from above. Slid down.
Has a great big giant chunk of limestone.
Anyone know how deep we were when we saw the bottom of it?
Oh,
How few many meters tall it is? 8 meters up. It says at least 8 meter tall. Localized mound. It's very like eroded on the outside, right? So it's like this massive rock with like lots of rounded edges.
mm-hmm.
Oh, nice.
Oh, that is nice. And you can see the layering in the. That's gorgeous.
Yeah, you can see that
So that's-
layering. Oh, that's neat.
Looks like the flat irons.
Look at that break in there. And then look, it's flat on this side.
Yeah, looks like totally like a slope failure. That just broke off and slid down.
Yep, turned sideways. We don't want to be around
I was
when
gonna
that
say-
happens. Avalanche of rocks.
That's exactly how I feel when I'm ever at the base of a cliff. It's like, well, this fell some other time. Why not today?
Right. Jeez.
I know, it's a little fatalistic.
That's really neat. We would like to collect that. Too big, huh? That'd be neat to get the aging on it, on those layers, and see how high up it came from.
Mm.
Reminds me of a ice cream cookie sandwich that I get at my local ice cream shop.
Right, right. This is a little harder, though. Probably not so good for your teeth.
Yeah, I got to be careful of that nowadays.
Yeah, I'm going to go to the dentist real quick.
Yeah, it's like just a big slab fell off.
Look at how the corals on top are growing sideways.
Yeah, I think we saw a slab like that earlier, and I don't know, it feels to me that, there was life on top that took the ride down.
Yeah, and how old is that, right? It couldn't have been that much long ago, because maybe they'd start growing up.
But there are some growing on the edge, too.
Yeah.
So, I don't know. Maybe they can only go one direction once they start growing.
Oh, right, like they don't bend.
I don't. Not for me to say.
Yeah.
Carolyn Rupel in the chat is commenting that this is referred to as lateral migration of the escarpment. The escarpment migrates back towards the plateau as these chunks fall off.
There's another flytrap anemone at 9 o'clock. There we go. Yep. Nice white hexactinellid sponge, looks like. And then we've got our flat mitten sponges we've been seeing everywhere.
Mm-hmm.
It's a rock migration.
Yeah.
And just a reminder, we're not at the bottom of this wall. We're sort of midway.
Middle of it, yeah.
So one can imagine that there must be a lot of this at the bottom as well.
What's the max depth here? 4,000? Over 4,000?
Of the area in general? Or?
No, yeah, the bottom of this wall, way down in the blue and the...
It's almost 5,000.
5,000?
Yeah.
It's
At the base as
approaching
well?
5,000, yeah.
Yeah, 4,700, 5,000, something like that. 2,100 meters high. That's
This plateau is?
the wall.
Oh my goodness. What did you say, 2.5 miles?
1.3.
1.3 miles?
Yeah. It'd be a heck of a hike, huh?
Yeah.
Gonna need more than burros to get there.
Reminds me of hiking from the sea up to on the big island of Hawaii, going straight up.
It's a long walk.
The ophiuroids we're seeing?
Is that more ophiuroids everywhere?
Yeah, I think so. There seems to be like one for every sponge, roughly. All over the place.
Ophioplinthacca?
Yeah. Plinthacca. Ophioplinthacca. There's a different yellow sponge here at noon. right on that rock edge. It's like right above laser. Yeah, I think that's different.
Oh, yeah, that really is.
Much thicker.
Oh, and there's another. Is this to the right to 9 o'clock as well?
Yeah, the same kind of like
Oh, I like them.
hand-shaped. Oh, neat.
Oh, wow.
That definitely looks like a mitten sponge.
Yeah, very cartoon mitten.
Yeah.
Yeah, there's another really well-formed one here. There are actually 3 or 4 of them on this rock.
Mm. Yeah.
That's cool.
Zoom in there. It's interesting that we haven't seen any fish.
Yeah. Not one.
I'm surprised.
Yeah, I mean, there's lots of habitat. There's lots of hidey holes and usually attracted to structures. My guess is a lack of food. That's really my first guess on that. Hey, who chimed in? There's a cool, this fan here, that's neat. We got a fan dead center screen. It looks similar to the flat mitten sponges we're looking at, but it's very large.
Also, we have Chris Ma's requesting a zoom on the opioids again when we have a chance.
Okay.
Check on the ID. The Kraken has eaten all the fishes.
So yeah, it was super steep wall. Probably 60-degree slope, 70-degree slope in the area. It's very steep. It looks like we're getting our flat mitten sponges are actually getting smaller and less abundant, right?
Yeah, we definitely have a downturn in the abundance of them. At least for now.
The boulders are smaller too. We had that one big, humongous wall boulder, or really chunk of wall that looked like it had slid down. And this is much. The rocks are smaller. There's more 10 centimeters to maybe 50 centimeters, jumbled, piled up.
Yeah, more rubbly, right?
Seems like the sponges are smaller too.
Smaller. The sponges are smaller, and maybe less abundant. I don't know. I'd have to sit down and count them all.
It's another bamboo coral right there.
Yep.
Maybe
Bamboo
another
coral at 9.
distance. I like that big sponge on the upper left, way off there.
Mm-hmm.
Another bamboo coral.
Nice. Now, is that, it looks to be the same, although that's short nodes, right, inner node right there.
Yeah, that is short there.
It's interesting how bare polyps this side of the bamboo coral is. See right at its skeleton.
Yep. It concentrates the polyps on one side.
Yeah.
Probably a feeding adaptation.
Yeah. It has those flexible little nodes in it, so it can wave in the currents.
Mm-hmm.
It's interesting.
Yes.
Look at how small the little space is from this node, but then it's
And it's big ones.
really a long one between the others.
Yeah.
You guys, there's an ophiuroid at noon, and 11 and 10 if you want to zoom on one of those for Chris. There's also some at 3.
They're all over the place.
Yeah. When you're done with
Take your pick.
There's plenty.
And Ken Sulak is commenting that, the lack of soft substrate's a good, there's a lot of critters in there that fish are eating. And so that lack can contribute to the lack of
like a fish.
fish.
Yeah. There's very long spines along each arm. You can see a really distinct central disc. That's to the upper left.
I think also Chris is hoping to get a view of the disc laid out.
Okay,
So
the top side of it?
Yeah.
Okay. We'll keep looking. Can't really get the right view on that for IDing him.
Should have mentioned that when I asked, but
It's a little flatter here, less of a slope in its immediate vicinity. And then as soon as we swing, you can see the wall drop off
yeah.
to the lower
Wow.
right. Just into the abyss.
Wow, I wonder if the current isn't so good here.
Yeah, it's much more barren.
Yeah.
What this little ball-like sponge, is that the hexactinella we've seen less abundant?
Don't know. We are too far away from me. But it looks like it's floating on top, like sitting on top of something. That white one. Oh, Hylanema?
It could be Hylanema. Yeah, absolutely. It's another stalked glass sponge. Again, that stalked form is present in a lot of different families.
Oh, that's the only one I know. That's the why I yelled it out.
That's okay. I think there's more than one. There's one right behind it as well on a stalk. Or is that... Yeah. Huh.
Cool. 2.
It does have a couple. And I wonder if that's just a different species growing on the stalk of the first one. Might be an ophiuroid laid out on the rock that's pretty close to center, yeah?
Oh, yeah, I see it, center screen.
Oh, yeah.
Get a good central disc view.
Ah, there you are, Chris. Oh, we have a little octocoral in the crack to the left as well. And that's nice. Look at that footage.
Yeah.
Seems to have a little damage on the tip of one of its 6 arms. 6 arms indeed.
Thanks.
Definitely the diversity picked up again real fast, right?
Yeah, it must be some kind of localized eddies or something that provide the, like it's probably in like an area that's like behind the lee. So wait, leeward side of the rocks.
Right.
There's that same similar yellow.
It just sort of
cartoon mitten.
yellow tinged sponge.
Yeah. Oh, neat. There you go.
And that guy's got 5 arms.
5 arms, yeah. Very floral central disc. Looks like a daisy almost.
You can see the dust. This camera's always so amazing. We've got some varied opinions on that stock sponge. You know, Nolan Barrett thinks it could be Amphidiscella,
Oh, neat.
which would be in a very different group than the Hyalinema. And again, so that was Ophioplinthacca.
This 5-armed one?
Yep. But the other one was weird, he says. The 6-armed guy. And so, I don't know. We will never know the identity of that stock sponge.
Well, there is one way to find out.
There is one way. Sir Collex-a-lot strikes again.
Sir Collex-a-lot.
If worn within reason, I'm going to wear it proudly. That amorphous sponge is also a big mystery. It would be.
There's another Hyalinema. Yeah.
Oh, yeah. Is that the same one?
Okay.
Is it easy to collect, or is this a nut? Oh, I wouldn't mind. I think it would be great.
And this is going to be sample number 6. Is that right? No.
I feel like I'm on 4. Yes, okay, good. And this would be an unusual morphotype, but we haven't seen this much yet. Kind of like those small guys.
Behind it.
Yeah.
There's also this yellow round ball sponge there. It looks like there's 2 above us.
Oh, yeah.
Up there.
I don't know how long this stock is.
Yeah, if we can get lasers for a second, that'd be good too. There we go. So maybe 30 centimeters?
Yeah.
Maybe 40 centimeters? So it looks like the bulb on top is about 10 centimeters, and then the stalk is maybe 30 or 40 centimeters tall. Maybe 40 or 50, actually.
There's a lot of happiness and cheering going on in the chat that this is going to be collected because it does have an unusual. People are commenting on the slender nature of the stalk. Yes, and everyone is very confused. The sponge people online are very confused about that soft sponge we've seen several times. So If that's another potential.
Yeah, let's see. Oh, look, we have a strawberry coral
Oh, nice.
at 8 o'clock at the edge of the screen. You can
This-
see the Anthomastus.
And that other stock sponge behind it, that's definitely a different morphology. I
Just a
believe,
couple inches?
yeah, just a few inches. This should be fine. Maybe we could image the base of the stalk just so that that's...
Like a little flower blowing in the current. There you go. Not much to see.
No, not too much to see, but anyway, it's nice to have. Just
It's almost like a
begin to-
brown layer at the base.
Thanks for that.
Yeah. You're probably going to need to use your cutters.
Hey, Stephanie.
Hey, John, what's up?
What are at the base of this Hyalonema type sponge? You see these flat bladed brown things sticking up off the bottom. What the heck are those?
I think those are the dead flat white sponges. I just think they're dead. Right? So they're that yucky brown of the white
There's
sponges.
a whole bunch of them all over that rock.
Huh?
There's a whole bunch all over that rock, like little flat blades, about 5 inches tall.
Yeah,
Dark brown.
I'm pretty sure they're dead versions of dead individuals of the white flat sponge that are next to it. Because they're the same shape, and they're pretty close to the same texture, except they have that, that dead sponge, funk on them.
Actually, Chris Diaz thinks they might be a different Facalia.
Oh, okay.
Yeah, and that they're, so I think she's agreeing with John there. And Tina Molodovtseva suggests that we may have seen some clatorhizids in there, in and among the stalks of the glass sponges.
Oh, nice. Clatorhizids, again, are carnivorous sponges.
Yes. They were small and only visible when we were all zoomed in there. Look at how delicately that's being held.
It's like this giant machine.
Well, it's a treasure. It really is a treasure. Going to starboard inner. Nice.
Starboard inner, number 4.
And I think we should go with Amphidiscella on the name for this one for now, pending. That's what Nolan Barrett suggests.
Yep.
Thank you. Thank you for that collection as well.
All right. And collection?
Absolutely. That's fourth collection. I believe that we must be getting about halfway through our transect, and we might want to consider a water sample.
Sounds good to me.
We're at about 3,622. So we're making our way slowly once again.
Yeah, when you're ready.
Yes, please. Speed Niskin bottle 3.
So it's going to be sample 5W.
That's what I have as well. Got to keep these numbers straight.
I know. Oh, look, we got some sand ripples here.
Yeah, that's
really
right.
Narrow channel there.
It's that guy. No, 9 o'clock. 9 o'clock.
Bushy Gorgonian of some kind.
Gorgonian?
Mm. My guess from this distance is going to be bamboo coral, but get a little closer.
More stock sponge back there, maybe? Oh, that's nice.
Yeah, bamboo coral. Again, with this little, like, cartoon mitten sponge, his yellow one.
That's right. That.
Another Hylanema-ish.
Yeah.
Oh, did you guys snap the Niskin?
Yes. Thank you. Just a little, I was a little late capturing that, but it's all right.
There's another large fan right dead center screen. Probably a bamboo. Very planar. So we only have about an hour and a half left. I think it'd be priority to move shallower.
Yeah,
now.
if we can move up.
See if we have any more species. And you can see the waves are lying parallel, perpendicular to the wall, so the the current is actually going across the face of this.
Mm-hmm. Someone just call in?
Hey, Alan, Scott Francis, checking back in.
Hi.
Welcome back.
Thanks. I'm almost disappointed to hear there's only an hour and a half. It means we're not going to go long enough to get into my invertebrate zoology lab this afternoon.
Yeah, unfortunately, the deep...
I just realized I could probably play it back on YouTube, on CTube.
Mm-hmm.
That's what I'll do. Looking nice, what I've been seeing in the background.
Yeah, it's been very high, a lot of sponges, high abundance of sponges.
Mainly the one species, but we've seen a few other, like round ball ones and a Hylanema lake that lives on a stalk. Also this like almost cream-colored yellow lobate cluster of lobes.
I had suggested at the beginning of the dive that it be a sponge phantasmagoria, but I think I heard you say it, Stephanie. Heard you say it earlier, Stephanie, that the wall, at least where we are in the wall, it's not as steep right here as what I was expecting. And when I referred to those sponges down off the Bahamas in the Little Apical Canyon, much steeper relief.
Yeah, we're on like a shelf part, and it looked like a huge chunk of rock had fallen off, and I think it's just sticking out in this area. I think once we start making our way up the wall, it'll get steeper. But it's like a jumbled pile of rocks here. Mm-hmm. Yeah. Yeah, we want to make our way up as, I don't want to say quickly, but, you know, as reasonably as fast as you can. Try and get as high as we can on, before we have to get out.
Yeah, I remember when you said that before.
I know, it's such a crock. Like, I don't even know what to tell you. Like, who am I kidding? There's, like, so much stuff to look at. Here's, like, one of those cool ball sponges with a large oscule.
Mm-hmm.
Geology, would you like a geology sample?
I have asked, but no one has made a request.
Yeah, go ahead. Yeah. Fire drills? Okay. Yeah, we're going to have a fire drill. Oh, don't. Oh, sorry. You're going to hear alarms, huh?
Very nice.
This is the drill. This is the drill.
Yeah, it looks like...
It's a nice gray sponge to the left and right of that white one as well. Sort of a charcoal-looking color. Oh.
It looks like we have a 3 stemmed gorgonian at 9. And then another one of those large cream-colored sponges with a large oscule. This might be Visalia. Did you see that? Yeah, there's a nice gorgonian. We got the yellow sponge with the lobate fingers. Stalked. Do we have an interest in that? Are we going to think about collecting that one?
Actually, yes.
The sponge in front. Yeah. Hey, Scott.
That's a primnoid octocoral.
Primnoid, okay. What is this thing? That's like a hydroid or something in front. What is this?
Well, let's see if we can get a little zoom in on that.
Down at, yeah, that guy. Oh, how neat. It's teeny tiny.
Wow, and it's
Baby sponge?
Baby sponge, maybe. Look, it almost looks like it has eggs or something in it.
It
like
does.
A circle.
Sure.
I have no idea what.
Claterize it.
Oh, that's a
Claterize
claterize. Yes, I wonder if it. Don't know if they brood.
I don't know.
Oh, that's a really
That's
nice claterized sponge.
Different one than I've seen. I've never seen that one.
Yeah, is that a
It's
form?
tiny.
Well, I've seen what I call like the flower or sunburst forms before, but I can't speak specifically to this one.
Just hoover up all the sponges on this.
Yeah, right. I think this might be Visalia, or a relative of Visalia. See the two? There's two of these ball sponges that are really spiky. One at 3 and one at 6.
Roselids.
Rosellida, yeah.
Oh, I get that soft mitt.
Yeah, I think the one right between laser is perfect. If you can get that one. Is that the one we're looking at? Yeah. You're calling that hexactinella anything better than that?
Beyond that, no.
Okay.
Look at the base. That's really interesting looking.
It is.
Yeah, we have. Let's see if we can get this done.
5-6. This is going to be sample 6.
6B.
This is very small. So it's less than 10 centimeters. I think they're all about that size, so
Yeah.
you can
Get what
grab
you
it.
can, and yeah, it will be helpful with the video for the overall form.
It's either going to be very soft or very crisp.
I think it looks soft, right?
It does. But sometimes you just need to poke it to figure out. Yeah, he's pretty soft. Oops, and he's got some. Oop. Awesome.
We
Okay.
definitely got some though. That's great.
Used a little bit when you squeezed him.
Thank you.
Cluster of lobate fingers in a fan shape.
And unless we have any refining on the identification, I think we're going to have to stick with class.
Yeah, right. Here's looks like a different species down here at 4 o'clock. Maybe a feria or something?
Yeah, farayid. And that's going in.
Yeah, be careful with port outer. It was one of those fan-shaped ones. It might be a little floaty. And he's in the box.
Nice,
All right.
really
Thank you.
nicely done. Again. The Farayid, we've asked for Zoom if we can get that.
Okay.
Mm-hmm. There's a lot of sponge-happy people in the chat today. So the Clatoriza that we got really good footage of, that looks similar to a Bicicladia.
Bicicladia.
Yes, known from the abyssal depths off Bermuda. Great.
And if you can, before you run off, can we look at that sponge that's now at 8-ish? 7 o'clock? Yeah, snap zoom. Oh, neat.
Much appreciated.
Definitely hexactinellid.
Yeah,
Looks related to Faria, but
yeah,
not
I think
right.
it's a Farayid.
Do you think so? Okay.
Yeah. Yes, thanks very much for that snap zoom on that sponge.
Thank you. That went on which one? Port. That was port inner, right? And that is number 6.
Yep. Just a time check. It's about 10 minutes after 1, and we are going to stay on the bottom until about 2:30. So we have an hour and 20 minutes or so left on this bottom time on this dive. On the wall of the Blake Spur, sticking off the Blake Plateau, down still below 2 miles below the surface of the Atlantic Ocean.
Yeah. You know, we can make our way a little more shallower when they get balanced. I think they're is your claw stuck open? Is that what you're struggling with? Okay. So that might be the end of our collecting. Well, maybe. Oh no, because they'd still have to pick the hose up with the same claw. So if the jaws are stuck open, then that's something that's got to get fixed on the surface.
Understood.
So we might be done collecting for the day, maybe. We'll see.
Other than the water samples.
Oh, yeah.
But yes. You missed.
That little hermit crab? Or
Yeah.
That's cool.
We can confirm that that's
What, the
that
claw?
the arm won't. Yeah, the claw will not close. Got it.
So it looks like we have an increase in those flat-bladed hexactinellid sponges again. They're really abundant in this spot. I would imagine it has to do with currents. The depth doesn't seem to matter too much as far as the abundance goes, just from visual observation.
Right, and the temperature has stayed pretty same at about 2.3 degrees
Oh my
Celsius.
goodness, it's cold.
So yeah, watch it just to be clear. We're just not using the starter for the water.
Yep, no worries.
Got it. Yeah,
So we might be done collecting biosamples.
I really like that cladorized sponge,
Yeah, that was neat.
carnivorous one, Abyssocladia. So there must be some animals around for that, other animals around for that carnivorous sponge to be eating. The way they feed is they have these, they use their spicules almost like Velcro, yeah, and they catch
They
upon
snag, right?
small crustacean larvae, that type of food, and then they migrate their cells over top of the prey items and
Oh.
digest them that way.
That's so weird.
It's a very slow way to go, I'll bet.
Yeah. They're so weird. So neat.
Yeah, you know, it's these animal phyla. They often have a what we call a body plan or some sort of like bow plan that they don't typically lose, but there are instances where that's the case. And for those cladorized sponges, the fact that they lose the water vascular system of that's typical of sponges is kind of interesting.
Looks like this white sponge is different too. This little like
Yeah.
a finger kind of branchy, and then there's this white ball,
Spiky ball.
Yeah, large oscule. I'm not sure if you guys can see that ashore, but on the left side of it, you can actually see the oscule right where the detritus is at.
Mm-hmm. That looks like another rosellid, huh?
Yeah, it looks like the Vaselia or something.
Or ally, maybe though. Yeah, we have confirmation on likely Rosellidae.
Alan, I think on that sponge there's an amphipod stick, that little, well, we moved out, but you could see a little filament waving back and forth, and there was an amphipod on the tip, and they usually construct these little filaments out of mud.
Oh, nice.
Oh, that is cool. The diversity within amphipods is
Oh, unbelievable.
it's
I can't
incredible.
even with that.
Like little insects of the marine realms. They just come in a lot of shapes and sizes and different niches.
That's what I was lecturing on in class today.
Oh well.
I wanted to follow up on your carnivorous sponge comments there. I was reading something recently about how these sponges digest. As you noted, it's very unusual for a sponge, which usually uses the plumbing, the aquiferous system. And the fact that the cells migrate over and enclose whatever they've caught. But the question is, does the sponge then secrete some enzymes that begin to digest that prey? And apparently the studies that have been done suggest no, they don't. Instead, they rely on bacteria that are also in there. The bacteria break down the prey, and then they would just take up the digested bits the same another sponge would.
Yeah, it seems such such a strange reach, like why make that modification evolutionarily?
Well, that makes sense because the other concept would be that they're basically forming a transient stomach and secreting enzyme, but that would be a big leap.
Right.
So what it's saying is no, they're not that. They're creating a transient space, but allowing the bacteria to break down, or just the aging of whatever they catch, they wait for a very long period of time. It breaks down, and then they just absorb the products.
Yeah, the sponges have a rich array of bacteria that they have inside their tissues and actually inside their cells and stuff.
I'm very glad I don't have a transient stomach. That would be pretty horrifying.
Well, we rely on a lot of bacteria in our own biology as well, but
my stomach stays where it's supposed to stay.
Sponges are particularly good at that. What was back there on the back?
Looks like it's another species growing there. Ooh, pycnogonid maybe? Yep.
There it is. Yep, it is a nice
cream-colored pycnogonid.
Sea spider.
Sweet.
Bacteria are partners, and especially the partners of sponges.
Yeah, you can see the current movement from our thrusters as we reverse. These sponges are just crazy abundant right here, so it must have really good current flow. It's just a great habitat for them. There are these. We collected one of these earlier, but they're really just
so abundant.
Yeah, they're really everywhere, and really every depth we've been at too. So there's only a few spots where there was no life at all, really barren area. For the most part, these are the most abundant species we have. It looks like. They were really crisp when we picked one up. It just broke right in half. And I don't see a lot laying flat, so I wonder if the current never really gets too strong in this area. Because I feel like if it was strong enough, it would just snap those all in half. And they'd be laying on the dirt. The sediment. Boulders are really large here, 5 meters long, maybe. Large, cobbled, like jumbled pile. And we have a very flat area along this wall. Okay.
Understood. Little delay while we gather.
They organize the ROVs to be together along with the ship. So they have to do ship moves, and then the ROVs kind of follow behind. So every so often they have to adjust the lineup. Like this giant boulder here with these same white, flat, low bait fans. They're sponges, hexactinellids. Again, we're seeing a lot of these ophiuroids too, the same pink, similar pink, long armed.
So I'm rechecking on the class level identification of that common blade-like sponge. Seemed like Chris Diaz was potentially referring to it as a species of Ficalia.
Okay.
And I'm not sure if she was talking about that one or the sort of tan, similar but smaller blades.
Mm-hmm.
Look at that rock.
Yeah, there's a lot going on this rock, huh? Yeah, so this little like stem here looks like a used to be dead or is dead stem of a coral possibly with a ophiuroid on it. And then most of the other stuff is a sponge. Ooh, that's a clatorized, is it not?
Yeah, the little branching thing. I think that is a clatorized sponge. Just a different form.
Mm-hmm.
Really cool.
And we got these like flat, I don't know what these are, these flat sponges encrusting. Yeah, there's the flat ones. They have like a veining, lots of veining. There's an oscule in the center. And then these brown, I think these are just this dead hexactinellids. The brown version of them.
Yeah, I think they're the dead form of the white one that's so common.
Another Faria, like folded hexactinellid sponge.
Still, as expected, not quite making our way up to the top.
Yes, it's hard to maneuver.
Well,
the straight wall.
Hard to maneuver, and it's a lot every time we want to stop and take a look at something.
All right, and these ROVs are operating 2 miles away from the boat.
Exactly.
The jumbled rocks are really common. That's basically what this habitat is this jumbled piles of these giant rocks. We've spent most of the time on this kind of outcroppy, flatter area. I think once we hit waypoint 2, it should flatten out and become more steeper, yeah. But we haven't gotten there yet, so we're just waiting on the ship move and.
one day in the future, it would be interesting to come back here to look to see, is there a difference at the depth that we're looking at right now between the more direct flat wall and this little sort of alcove that we're in? Because this may be somewhat, it's either protected a little bit from the currents or it creates a little basin where you can have some swirling currents. And I just wonder if that might make a difference to whether we're seeing a whole bunch of sponges or the morphology of the sponges we're seeing, where a lot of these delicate ones versus more robust sponges and corals on the vertical wall.
Right. So that requires additional exploration. But this creates the baseline.
Right, exactly. We keep getting these baselines, and we're all eager to come back and ask all kinds of questions that have been revealed from our little site.
Yeah, it looks almost flat right here. Take a zoom in there. Yeah, I think that is maybe a pen on the back side of that rock. Ooh. Yeah, I think that's a rock pen, but hopefully Scott will chime in if that's not the case.
Definitely a sea pen, Alan.
Is it down in the soft sediment?
I can't tell right now in the sediment or if it's on the rock.
Right. See it.
It's a pretty deep one, I'll tell you that. Yeah, you know, I'm not 100% convinced that was a rock pen.
Oh, okay. And is this another Metallogorgia here?
Now, this is a Chrysogorgia.
The Chrysogorgia. Oh, look at it.
Yep, same family.
Yeah.
Chrysogorgiidae. You can. They often have those sort of gold-tinted skeletons. This one's had a hard life. It's lost a lot of branches. Some of the tissue is scraped off down below, but you can kind of see that gold tint to it. And what has it got? I guess it's got a bunch of anemones there in it. There we go. Yeah, that's a nice view of the underlying gold skeleton. Then you can see the very thin layer of tissue. Chrysogorgias always have a very thin layer of tissue, and the polyps are like little snowflakes or drips of water on the branch. Very delicate. Yeah, I think I'm seeing a few anemones overgrowing in parts, an amphipod.
Mm-hmm. It's a nice little home.
Yeah, the Chrysogorgia are also very frequently home to shrimp and chirostylic crabs.
I'm sorry, could you repeat that? I'm sorry, Scott, I missed that.
Sorry, I was looking at the screen, so I'm
Sure.
not talking down to the phone. I'd say the Chrysogorgia colonies are often homes to shrimp and chirostylic crabs.
Oh.
Not this one, but it's very common. I'd say those are ring anemones, but Daphne Fountain wouldn't like that. I don't want to dishonor her memory.
That is sad thought thinking of that. Daphne Fountain, who is a fantastic anemone specialist, passed away this past year.
I'm sorry to interrupt. We get a close-up on the white branching coral to the right. Excuse me, to the left, I'm sorry. At like 8 o'clock now, 7 o'clock. It's like slanted with several branches going up.
Yes, this is it.
Thank you. That's not at all what I thought it was. Okay, that's Primnoidae. Maybe a Calyptophora. You can actually nicely see the scale-like sclerites on these polyps, and the number of those scales and the orientation of those scales help to put it into a genus. There's something weird going on the right side there. I see, looks like a hyperinflated polyp. Not sure what that is.
Oh yeah, maybe we're looking at the side of an anemone that's pulled in.
Maybe, yeah.
It's
weird. Okay, move on as you're ready. Thank you.
sure. There's quite a nice anemone to the left, I believe. Let's see. Yeah, up and yeah, maybe take a little close up of that guy. Thank you. Nice. He's got the large mouth sitting there waiting for food. Thanks a lot.
Another cladorizid sponge just in front of it.
That's right. The same branching form we saw before. Good. Don't want to run into any obstacles. It flattens out here. You can see real increase in the amount of sediment, sand and silt on the bottom.
for certain, but I'm guessing that these unbranched whips that are fairly straight are in the genus Bathygorgia. They are bamboo corals. And
Thanks, Scott.
Bathygorgia?
I had to pause there while somebody was wheeling a cart outside my office. So most of the bamboo corals, when you hear their names, have an isis associated with them. So this is one of the few genera that does not, Bathygorgia. That name, Bathygorgia, refers to very deep Gorgonian. And for a long time, it was the deepest known of the bamboo corals. But on these Okeanos Explorer expeditions, we've found other deep ones at about 4,800 meters that are in the J clade. Haven't yet been described, so we don't know exactly what genus they are.
Oh, nice.
But that's my best guess for these. And they often do have the polyps oriented on just one side of that stalk.
There looks like a fan coral. Let's see, 4 o'clock of laser, so down and to the right a little. It's kind of a weird angle. We can look at, yeah, there you go, right there.
Brushy and bushy.
Center screen, yeah. There you go, right there. Have we seen that one yet? What is going on here? It looks like it's growing off of this one of the dead sponges. That's interesting. I have no idea. What do you think?
Wait a second. These
Sure that's not a hydroid?
those polyps look like hydroids.
Hydroid, yeah, maybe.
Absolutely.
The stylaster?
I wouldn't
think it's
expect
not
the
solid.
polyps to be so large.
Yeah.
Oh wow!
I think I'm going to get a name for you. Just give me a couple minutes here.
You know this one, Scott? That'd be great.
Oh, yeah, I wouldn't go so far as to say I know it. I recognize something like it. And I'm going to see if I can find the name.
Yeah, this looks like a calcified hydrocoral, but
I
not.
completely agree. This is a hydrozoan.
Yeah, not. Oh my goodness, I love it. But not stylasterid. You can
So is
see
the-
how much larger these polyps are. They're really fat and chunky.
Is it a colonial hydroid? It looks like it's
I think these are related to hydroctinids.
So like it looks encrusted, right? So
Yes.
the bottom part is encrusted. Is this branching part of the hydroid, or is that something that was there before that it's then infested on? Right. So it's
I think that it's made that branching
shape?
skeleton,
Okay.
but I'm definitely not sure.
Okay. That's weird.
Oh, for my claw! Oh, not my claw. No, that's just really, really beautiful and something we haven't seen yet at all.
So I just typed in the chat hydrodendron. That's the name I was trying to remember. I don't know. I don't even think that's what this is, but that's the name that I had in the back of my head. And I'll just look around a bit, poke around a bit on the Benthic Animal Guide, see if there's anything.
Thanks. We'll be able to get at least a generic ID from that great footage. Oh, look, the whole thing is just really unusual looking. Oh, it's growing off of the
rock,
and you can
too?
see that it's making branches off the rock. Can we check it on the rock just to the left? Thanks.
Yeah, it looks like almost like it's an encrusting. It seems to encrust the flat parts, and then it grows these branches up.
Yeah.
So it's, yeah.
That's what they're doing everywhere, all over. Only usually they're much smaller. Thanks. So it has a stolonal mat, and then it's growing up from that.
Really weird.
Really weird and really nice. Thanks for that. We can check for something new now. Well, that was a real treat.
Mm-hmm. So we still have these piles of large boulders that look really rounded this area. They look sharper a few minutes ago.
Mm-hmm.
But here they look super rounded, like they've eroded down. Is that a different star here at center screen? Right there on top of that rock? Oh, neat. So I've seen this genus before, and yet it is escaping me. Where are you, Chris?
Oh, that is nice.
Brisingid, thank you.
A Brisingids.
Mm-hmm. Looks like a chitin maybe at 3, too, right behind it. That's different. Possibly
I was going to ask, what is that below there on the rock?
Yeah, it looks like it could have been a chitin, but it's so maybe it's a worm.
Yeah, I think it might be a scale worm.
Maybe?
Oh, and there's more of that hydroid growing there.
Well, that's what your eye would go to.
A scale worm. Nolan is saying scale worm.
Yeah. Wow, it likes this depth.
Hey, this is Chris Ma.
Hey, Chris.
Hello. So I'm looking at your Brisingid. It's very delightful. This looks like Frayella, which is one of the more common Brisingids that we see at this depth. Frayella is named after Freya, the queen of the Norse gods. All of the Brisingids have, for whatever odd reason, well, there's a distinct reason. They're named for Norse gods. So because of the myth that Freya was, so the original Brisingid name is based on Brisingamen, which is like a necklace or a breast ornament, and Loki steals the breast ornament and throws it into the abyssal ocean.
Oh,
and
neat.
so all of the names tend to come from different figures in Norse mythology. That's changing, but it all goes with the myth, the story of the original name. So Freya, Freyastera, et cetera, et cetera. The little worm that you saw is a scale worm. They are actually associates of Brisingids. There's a couple of species accounts where, lots of asteroids have scale worms as associates of they live in the tube foot grooves, and I've literally seen preserved specimens that have the scale worms still in the tube foot grooves. They are very close. They have a very close relationship.
Oh, neat.
I don't know necessarily which one that is, considering all of the ones that I've heard of. It's probably new, but I've definitely seen in a lot of the pictures scale worms that live like just away from the animal, or possibly like on the underside where the tube feet, where the tube foot would be located. Probably just, I think the word was kleptoparasitism. They go in and look for food as the animal's feeding, because Brisingids hold up their arms in the water and capture small crustaceans with the spines, which have lots of little claw-like structures called pedicelaria, what I've referred to in the past as the Velcro of death. So there's a whole thing going on there. It's always fascinating, and this is the first Brisingid we've seen on these cruises for this expedition. So I'm always happy to call in and go on and on about them. They're the first animals that
We're happy to have you. And I think we've got a little Medusa floating by.
Thanks, Chris.
Uh-oh.
Oh, no, that's a comb jelly. Yep, absolutely.
I'll talk to you guys later.
Thank
Bye-bye.
you.
Bye. Yeah,
Comb
nice
jelly.
low bait comb. Probably Bathycyroe.
I also saw a candelabra-shaped fan around 3. Yep, yep. Oh, I guess now it's center screen, yeah. Oh, Scott, I know you know what this is.
No, this one's not a bamboo coral.
Oh, really?
Yep.
Nice.
Gonna be Cryptophora, I think.
Cryptophora.
I believe so. I will verify that it is that genus, and I will type it in the chat for you.
Thank you.
But it's definitely family Primnoidae.
Primnoid, okay. Thanks, guys. Looks like a lot of increase in sand. Here you can see the sand waves. So that means that the current is going across our view based on the sand waves location. There might be a giant fan in front of us. Yeah, it's a super weird rock. Oh, damn. Tricky. There is a different yellow sponge here in center screen.
There is a different yellow sponge here in center screen. There is a different yellow sponge
Hey, if you guys could mute your phones if you called in.
There is a different yellow sponge here in center screen. If you called in. There is a different yellow sponge here.
Science, I'm going to separate the phone call for the moment.
It looks like we have another yellow crinoid we had seen earlier at 3.
I like this view as well.
It's a good garden. Nice. So that's another Cumachuli crinoid. There's sea lily. Maybe about half an hour left on the bottom. I see another Chrysler at, 3 o'clock? Maybe another Rosility at 6. But otherwise, most of these white low bait fans are pretty much everywhere. Yep, Chrysler. That's amphipods.
Oh, nice.
And pink polyps. You can see the zigzag stalk inside, where all the branches come off of. That's pretty neat.
Yeah, something about that zigzagging pattern. One of the things about Chrysler and the way that the genera are defined, and the species actually, is that there's a very regular geometric pattern to how the branches arise and how that stalk kind of zigzags its way up, such that if you held the colony on end and looked down on it, you could count the number of branches that occur in one rotation to where a branch comes to overlie a branch below it. And so you determine, is it one rotation, two, or three rotations, how many branches are in that rotation, and then is it rotating to the right or left. So that tells you something about how regular the pattern of growth is in these different colonies.
Oh,
it's really
cool.
quite incredible.
Looks like a huge bamboo coral ahead.
Wow.
It's like a meter tall at least. There's lots of hairy stuff on this rock here, on the edge of it. Yeah, fuzzy rock. I can't really tell what it is from this far away, but it might be gorgonians or hydroids. Definitely those white sponges. Yeah, looks like a bamboo coral, planar, sparsely branched.
Brittle stars dotting here and there.
Of course.
Almost assuredly, this bamboo coral will be in the J clade, which means it's in a clade, a genetic clade, a group of species that are all related to the genus Jasonisis, the only one that has currently been formally described in that clade, and therefore we give it the name J.
Mm-hmm. J clade bamboo coral.
A lot of the bamboo corals in this clade are characterized by having a kind of a milky appearance to the tissue, and that's because they have very small scale-like sclerites embedded in the tissue that reflects the light, and so they're not as transparent as some of the others.
Oh, neat. Looks like they have nodal branching, too, where the branches arise from the nodes.
Yeah, I couldn't tell, Stephanie, if it was the node or
just a hair above it.
Past the node. Yeah, I think you're right. I think it is the node, but it's close. It's interesting there. Is that like a trifurcation at the base there?
Yeah, I was just about to. Can you guys look at the base for me when you get down there? It looks like there's 3 there, and that looks like 4. And they all look like they're on the node. At least 2 of them are on the node.
Well, that's interesting because it looks to me like it's not planar, which is also important to know. Is it planar or is it forming like a 3-dimensional bush? And that would be more like a 3-dimensional bush, and that's definitely nodal. That's what you would expect from Acanella. This is not the genus Acanella, though.
Oh, nice.
Good. That's a good observation. Thank you.
Mm-hmm. Looks like we have another bamboo coral at 3 o'clock. And then one dead center screen that's a single stalk with an ophiurite on the top. Yep. Looks like the flesh on the top of that stem is all worn out. It's gone. Yep, exposed skeleton.
Yeah, so that's interesting. We often see the reverse pattern where the the tissue at the base of the colony is gone because a sea star typically has been feeding on it. But this is the reverse pattern, so it makes me think that it's the abrasion from the arms of that brittle star, perhaps.
That's caused that sloughing of the tissue?
Yeah, I mean, it's a hypothesis.
That's neat.
There's certainly plenty of spines in the arm of those brittle stars.
Right. Hey, Nav, is this science?
No.
Oh, I see a star. Yep. Yeah, right, just 3 o'clock of laser. Right there, center screen almost. No, it's a little bit 4 or 5 o'clock of laser. And it's on the laser now. It is teeny tiny, yeah. At the size of our laser dot.
Good eyes.
Looks like a 6-arm from this distance. Yeah, teeny tiny guy. He's poofed up.
You see 2 stars in a right.
Like 6-arm star.
2 stars.
Is it 2
Oh,
stars?
7 are.
Isn't this a star too? Underneath it?
But is there another arm just to the going off
Maybe
to the left?
Maybe a 7-arm, yeah. Looks like it.
But thanks. Yeah, it could be two stars.
Down.
Okay.
Yeah, sir, but like half of a pinky nail. Tiny bit of nothing. You can see the tube feet coming out, the one on the bottom.
Yeah,
right
out
at center
to the
screen.
left hand.
And it just, I can only see 2 of his arms. I think the rest are probably tucked under. Chris Ma?
Hey, this
Hi.
is Chris Ma. Hey. So what you have here is some kind of solasterid. You've got me juggling books here, so pardon me. So this is the members of this family are predatory. And yeah, so this is actually really exciting because you're seeing 2 species at war, so to speak. One is eating, one is trying to get away. Because that lower one is a pterasterid. So the lower one is a slime star, or what I've been calling, so it's a pterasterid. I'm not sure if it's producing any kind of defense at the moment. To be honest, this is the first time I've ever seen one star feeding on another of these 2 groups. And it had always been presumed in shallow water species that the defenses of slime stars were directed at predators, such as sea stars like this one. This is kind of double duty because I'm trying to. So the predatory one that has 7 arms, that one looks like it might be something called Chrysaster, but This is really deep for Crowsaster, so it's probably not. It's probably something else. And the other one is more like I said, like a pterasterid. To be honest, I'm not sure which one it is right off the top of my head. I'll have to sort of study these images a little bit later to figure out which one is which.
Awesome.
Did I hear earlier that collections are sort of off the table for the rest of the dive because of technical issues?
Yeah.
Yes, you heard correctly.
Yes. Okay. Well, in any case, simply the observation of this is really a banner day for me because, as I like to make the point in other dives, sometimes just watching these ecological interactions is more important than having yet another new species because it tells you something about a lot of the biology that goes with these animals that we know only from specimens. And because a lot of the historical record for these specimens comes from trawls, and the trawls, of course, collect the specimens jumbled together like, parts of a blended smoothie. You can taste them, but you can't quite tell what they were doing beforehand. And I've written whole papers about observations of these sea stars from prior Okeanos dives.
Oh, nice.
And this is probably yet another example of that. So, I will probably be downloading this video from the Okeanos server whenever it's available, because that was pretty exciting.
Great. Now we're going to zoom in on
and
some.
if we see it again, I'm sorry?
No, just commenting that we've got something exciting on Zoom right now.
Chris, before you run away, can you put those two names into the chat for me?
Sure, I will.
Thank you so much.
Thank you very much, Chris.
Is this a red hexactinellid?
Okay. Bye-bye.
Bye. Are you at a loss for words?
Yeah, I mean, I do think this is a hexactinellid, but I don't think I've ever seen anything this
weird.
Yes, this weird, this color. Very beautiful. So these are...
It's like hot pink. It's a bright pink color. Pinky purple.
I just keep trying to figure out if it's the source is somewhere else, but it really looks like the red is permeated all throughout the sponge skeleton. That's nice.
Yeah, it has the same texture.
Oh, yeah. It's not a hexactinellid,
You don't think so?
I don't think so. I don't.
Poke it. Poke it. See what it does. That's so weird. I mean, from a distance, it looks like, like a red algae, but there's no way.
Yeah.
There's living red algae down here. It's way too deep. But when you get close, it has that meshy...
Nolan Barrett thinks it's a hexactinellid.
Yeah.
Just the colors are really throwing. Just never seen anything pigmented other than, yellow. It's just white or yellow. But Nolan is also leaning towards not hexactinellid after the close-up. What was that?
I don't know. Very odd. Oh, let me get... Hey, Nav, this is Science. How far away from waypoint 2 are we? Do you think we'll make it before we have to come up? No? Okay. There looks like another crinoid at 3-ish, 4 o'clock. A yellow guy. More bamboo corals. Maybe some Feria on this rock down here in the corner. And again, these white fan sponges are the most common. They're everywhere. Oh, I don't know. Not me. I think I counted every coral on Pulley Ridge for 5 straight years. I am not counting these sponges. Yeah. This is like a sand chute. Like waves of sediment. And then at the base of this like jumbled pile of rocks. And some outcrops, yeah. He just disappears into the background. You lose the light.
It's a big hill.
Yeah, right?
So with that red pinkish sponge, I think I got a little overly excited about it.
Oh, God. You're going to make me snort into the microphone.
Apologies for that.
Don't apologize to me. Apologize to the people listening.
I was.
Do we get a name for that pink sponge or no?
Chris is suggesting Hertwiggia.
Hertwiggia. Isn't Hertwiggia a hexactinellid?
It is a hexactinellid, and Chris Bronco, our postdoc at NMNH, is saying we do have a red Hertwiggia in the NMNH collection. So maybe it's not so maybe it really was a hexactinellid.
Yeah.
She did say that this one looks different, so I think she was just acknowledging that the color, that's not necessarily the Hertwiggia that we were looking at. So sorry, I misspoke about what she wrote.
Thanks, Scott. Well, now that's the perfect...
Yeah, type specimen.
Yes, exactly. This is the sponge you'll find if you come here.
Yep. It is the type specimen that needs to go into the museum that all other specimens of the species are compared to. It is perfect.
Well, if it's undescribed, the one we collected is going to wind up being the...
Well, then we should collect this one. Make this one the type specimen.
Too late. Yeah, collecting video, exactly. Well, having established a video as a type specimen...
That's neat.
It can definitely be done.
Yep.
That's really nice. Perfect. Nice little white guys.
Yeah, living on them at 9. Little commensal guy.
Yeah, can we check in a little zoom there? Yeah.
Yep.
Yeah.
Amphipod.
Amphipod.
Is it similar to the one we saw earlier that flapped its rear end?
No, that was a big isopod.
Isopod, okay.
That isopod was crazy.
Yeah.
I kind of was hoping we would see it a bunch of times.
No, I only saw the one, I think.
Just the once.
Okay. Large boulders are still here. They're like nestled in between these. Well, in between these boulders is pretty thick sand. It's got some sand waves. Boulders are what, 1, 2, 3, 4 meters wide, maybe meter, 2 meters tall. And again, these white fan sponges are everywhere. They're crazy.
So
another bamboo coral here at 4-ish, 5-ish. Oh, the bamboo coral we were talking about. I was talking about. Okay. Okay, thank you.
great. 9 more minutes on the bottom here.
So it looks like nodal branching. Looks fairly planar. A little pink hanger-on at the top.
Nice.
Yeah, look at
Red
that.
mouths.
There's a little
A
commensal guy, snail or something.
there, or maybe. Wow.
You can see the little pinnules on each of the arms, on the tentacles. There's a lot of stuff on this little rock. It's just covered in stuff.
Yeah, it sort of looks like just a really weird coral reef.
Yeah.
You know, like a bommy you're back in the little shallow bay, and only all the organisms are different.
Very Dr. Seuss-ish.
Yeah. And just... Yeah, that's... Mm-hmm.
Yeah, they're really rugose, but like eroded down, so like almost like they were at one point sharp, and they've just been beaten up from the waves or the wind. I mean, get the right word here.
Yeah.
The current going
the current
fast.
fast.
Yeah.
You can see all the sand channels between the rocks. One of my formative experiences as a graduate student, we had gone out and we were studying fossil reefs. So
cool.
I had never gone to a living reef before. I was studying fossil reefs and getting these lectures on this is, where the little channels are. And then when I went and saw a real reef.
A living reef?
Yes, it was like, well, click. Oh, now I get how this whole thing goes together. Yeah.
Like the spur and groove formations they made from the water movement.
Yeah.
They're pretty cool. Yeah, it just drops into nothing.
That's a nice view.
Mm-hmm.
Just the massive extent of this. I mean, we're just looking at one tiny piece of the wall of a feature that's hundreds of miles long, right?
Yeah. I'm not sure how long it is. Let's see.
Well, the outer edge of it should go from
like 2,100 meters is what we have written down, and that's for the vertical relief through the Blake Spur.
Right.
Yeah, it's huge. It's a huge feature. Yeah, it's very Grand Canyon-y. On the scale of the Grand Canyon. It's bigger, though. Deeper than the Grand Canyon is. Yep, and we're about 8.5 Empire State Buildings stacked end to end underwater. I think it's hard for, like, especially when we talk in meters in the United States, like, a lot of people have a hard time with meters because it's metric. And we always think in feet and, yards and stuff. So yards about a meter, give you an idea. But when you say it in Empire State Buildings, it's like, okay, now I get what that is. It's 8 Empire State Buildings. That's a pretty deep dive. Yeah, we're about 274 miles from shore on the edge of the Blake Plateau. This actually drops out into the Blake Canyon. Is that right? The Blake Canyon?
No, I don't think so.
Am I making up words now?
We have the Blake Escarpment.
Blake Escarpment.
Yes.
Yeah. And it drops down to this lulled area at the base. There haven't been really many dives here. There was one here from Woods Hole, it's Alvin. And it was at the base of this wall. And I'm not sure what year that was. So yeah, the Blake Plateau starts at about 450 meters deep, closer to shore, closer to the U.S. Shore. And then it goes about to 1,200 meters, so this like slow slope out to the east. And that ends at about 1,200 meters. And then out of nowhere, it just drops straight down. And that's what we're looking at right now is that wall, that straight wall. We're, of course, in this local area. We actually have some flattened areas, so we're able to sit on top of these features. But this wall here is really steep. The top of the plateau, right where we are, is 3,156 meters, and then we got down to 3,740 meters along about halfway down the wall. Oh, we need to take a water sample before we leave.
Yes, we do. And that leaves us one additional water sample to take on the way up.
Okay. Yeah.
Yes, I do. And this will be Niskin bottle 4.
Yeah, so this is NOAA Ocean Exploration, and I'm Stephanie Farrington. I work for Harbor Branch Oceanographic Institute in Fort Pierce, and I'm with my co-lead, Alan.
Alan Collins of NOAA Fisheries and the Smithsonian National Museum of Natural History.
And today we dove the Blake Spur, about 275 miles from shore. And then I think tomorrow we're going to do the deeper dive. Go ahead if you need to talk to me. Okay. It just went? Okay. We good? We had a successful one?
Thank you.
So that was our eDNA sample number, the 4th eDNA sample, which is actually our 7th.
7W.
7W, so 7th sample of the dive.
And unfortunately, we had to stop collecting specimens because our arm stopped functioning.
Mm-hmm.
The claw of the arm.
The claw's jammed open. Okay, so. All right. And the plan is to have our post-dive call now. Is that what Matt was talking about?
Yes, at 2:30.
Yeah. Okay, so in about 5 or 6 minutes, we're going to have our post-dive call, so feel free to stay on or call in if you want to have a say in tomorrow's dive.
I would like to do it about the top of the plateau, which I believe was about 2,500 meters. Thank you.
Good. Yeah. So hang on. We'll for those of you guys at Shoreside who call into the line, we're going to have our post dive call, our pre-dive call for tomorrow at 2:30 in about 5 minutes. So just hang out. Otherwise, it's going to be about a 2-hour ascent time to get off the bottom and recover the ROV. Good? All right. Oh, sure. So, yeah, so this dive to me was dominated by this white, I believe, hexactinellid sponge. You said you had a name for it.
Chetopleura is at least a possible ID.
Possibly Chetopleura. Yeah. We actually collected that when we went to collect, it was really this brittle fan, and I'm surprised by that because that means that the current isn't that strong here, because otherwise this would just collapse, I would think. But anyway, so we collected one of those, and we also saw the dominant fauna we saw were the ophiuroids, or the brittle stars, and then a few other species of hexactinellids.
Yeah.
Feria, there was a pink one that we thought was, which one?
Not sure about the pink one, although it might have been Hertwigia.
Hertwigia, so another hexactinellid. What else did we see?
We saw
2 sea stars eating each other, or 1 sea star eating another sea star.
Yeah, that was exciting. We saw a great isopod associate on
the Brazingid, right?
Yes.
Brazingid sea star.
And that had polynoid scaleworm associates with it.
Let's see.
We saw a calcified hydroid that was overgrowing rocks and other organisms, and
at least 2 different species of carnivorous sponges. That was cool.
Yeah. Abyssocladia, and then also a branching form.
We collected a Hylanema-esque stalked sponge.
And then we saw quite a lot of gorgonians of different types. Chrysogorgia.
Yep, and bamboo corals were pretty common. I think probably bamboo corals, the ophiuroid, and that white flat fan sponge are probably the 3 most common species we saw.
Right.
The rock wall was actually not too stark or sharp where we were. It was more like jumbled pile of rocks. We didn't actually make it up to the wall side, so tomorrow we'll probably see more of a stark, straight-up wall. I don't think we have. We're going to land on the bottom. But yeah, we were. Our max depth was about 3,740 meters, which is roughly 8.5 Empire State Buildings stacked end to end, or on top of each other. I would hate to be the engineer who has to design that. The shallowest point we got to was about 3,500 on the end of the dive before we had to pull off bottom.
3,600.
3,600.
Yeah. Okay. We didn't make a lot of vertical relief.
Yeah. Yes, that was really neat. Really neat. And the only other, I mentioned that earlier, the only other dive around here was from the Alvin. So this is totally exploratory.
It's really our first look at this Blake Spur wall.
Yeah, it was neat. That was cool. Thanks for joining us today. Tune in again tomorrow. Hopefully the weather, we don't expect the weather to be bad tomorrow, which is why we're planning our super duper deep dive. I think it's going to be the deepest dive of this cruise. But thanks for tuning in to Ocean Exploration, and we hope to hear from you tomorrow.
Yeah.
All right. And for our scientists ashore, go ahead and hang on to the phone. We'll be with you shortly.